So we are interested in the first replicating molecules, which could well have been RNA or something very similar. And we're imagining that there was something like a polymerase ribozyme, which in our, do we have a pointer? No. It's the red one? Okay, so in our cartoon, it's the red blob is a polymerase ribozyme, which is functioning as a catalyst. And it's binding to another strand, which is the template, and we're supposing the template is the same sequence as itself, and it's making the complementary strand to the template. And there are ribozymes that work like this in the lab, so this is a real one that came from the 2001 paper here, and the best ones of this type now go up to 200 nucleotides. So this is good, except that this one won't work with its own sequence as a template, so it doesn't yet replicate itself. So if we think about there's three requirements that a sequence must have in order to sustain replication in the RNA world. It would have to be able to work on its own sequence. It would have to be able to replicate faster than the breakdown rate of the same sequence by hydrolysis, and it would have to be accurate enough to maintain the sequence. And roughly speaking, that means that the average numbers of errors per whole sequence replication is one or less. So that's the error threshold criteria. The basic theory of the error threshold goes back to Eigen several decades ago, and I'm going to explain it on one slide. So P is going to be the concentration of my functional molecule, which is a red square there. It can replicate at a rate which is R0, and then X is the concentration of mutant sequences, which are black circles on here, and they replicate at a slow rate, R1, which is less than R0. There's a breakdown rate, which I'll call V, and I'm assuming for the simple case it's the same for all kinds of sequences. So in order to survive, the good ones must replicate faster than V. So the red ones have replicate faster than V. And then there's a mutation probability, M, which is the probability of a deleterious mutation somewhere in the sequence per replication. And it turns out then that there is a maximum error rate that can be sustained. That's called the error threshold. So the red ones survive if the error rate is less than the maximum M, which is called the error threshold. So this is a very simple theory. It's a 5-minute theory, and what comes out of it is this: if the replication is perfect, so M is 0, then I get all red ones. As I turn up the mutation rate, the red ones go down and down, and there's an error threshold here, which is the maximum sustainable error rate. And since in this case, this is the case where the black ones, the X's, cannot replicate by themselves. The replication rate of the R1, the replication rate of the black ones is less than V, so the R1s die when there's no red ones left. Okay, so everything dies when mutation is high. So that's the basic error threshold theory. But we're not interested. Well, I'll go back one. This would apply to something like a virus that is multiplying inside a cell, and the cell provides the magical ingredients that are necessary for the virus to replicate itself. But in the RNA world, there's no magical cell there, so the RNA ribozyme has to copy itself. So now our reaction scheme looks like this. A red one meets a red one, and it makes an orange one. The orange one is the complementary sequence to the red. If a red one meets an orange one, it makes a second red one. So together, by the two-step process, the red ones can replicate. But if a mutation happens, instead of a red plus a red should make an orange, but it makes a black one by mistake. And a red plus an orange should make a red, but it makes a black one by mistake. And if a red meets a black, it makes another black. So we're always assuming that mutation takes you from the functional ones to the non-functional ones, because it's much easier to go wrong than it is to put your mistake right. So we just assume mutation makes the bad ones all the time. And so now we can do a 5-minute theory of this. So these equations are the well-mixed theory. They just tell you what would the concentrations of these molecules be in a well-mixed reaction system. That's a 5-minute theory. And the 5-minute theory of this says everything dies because the parasites take over. In this case, the parasites always take over. And this is a cooperative system, right? one red one can't do it by itself. You need a group of red ones. They have to cooperate, and cooperative systems are taken over by parasites in the well-mixed case. So now we already know, because this kind of problem has been studied many times before, we already know there's two ways of saving you from the parasites. Either you have spatial clustering or you have protocells. So in a spatial clustering model, they have positions now. The molecules have positions. There's still three types of strand. And what happens is when you have slow diffusion in a spatial model, you get clusters of functional ones together. So the functional ones meet the other functional ones more often than by chance, and therefore they have a benefit, which is counteracting the mutational benefit of the parasites. So clusters of red ones can survive, whereas well-mixed red ones do not survive. A similar but different argument explains why replicators in protocells survive. It's because when we have finite numbers of strands in protocells, some cells are good. Here's a good one. It has only functional molecules. This one is going to multiply fast and divide and produce other protocells which also have functional molecules. Whereas something like this is being taken over by the black parasites, but this one is not going to grow and divide. So some cells are destroyed by parasites, but not all of them, and the whole system survives because there are some good cells which are not taken over by parasites. So that's group selection, right? So this is group selection, saying selection works at the level of cells as well as at the level of molecules, and it's the group selection at the level of the cells that favors the survival of the polymerases. So all that has been fairly well studied, but the problem is it's apples and oranges, right? What I want to know, then the question for this talk is, which of these two mechanisms is better? And by better, I mean, which makes it easier for polymerases to survive? So that means a lower minimum replication rate is required. In a better model, a lower replication rate is required, so it's easier to find a functional catalyst that works. And in a better model, the error rate that is tolerated is higher. It's a higher mutation rate, a higher error threshold, okay? So I want to be able to compare spatial models and protocell models in a quantitative way. And then we hit this apples and oranges problem because they have different parameters. So then we have to do a bit of thinking about what does a spatial model mean. So in the simplest spatial models, we put things on we put one strand on each lattice site and we say they interact with their neighbors. And so, well, maybe that means something like a surface, and molecules are stuck on their surface, and two molecules next to each other can interact with each other. But I find that hard to believe. I can't imagine real molecules doing this if they're actually stuck to a surface, right? If this is going to replicate its neighbor, well, maybe the neighbor needs to move somehow, so it can't be stuck. And if it's not stuck, then why does it not diffuse off into the third dimension away from the surface? So I can't really see these spatial models representing really ribozymes that are permanently stuck to a surface. But what makes a little bit more sense is to think about the spatial models representing molecules which are in a constrained environment so that the diffusion is slow. So what matters in these spatial models is diffusion is slow, so clusters arise. And diffusion in a 3D open pool is going to be fast, so you get well mixed and things die, right? So we have to have if the spatial model is to be relevant, we need an environment where diffusion is slow, and one such might be pores in a rock. So this is a paper proposed a while back by Koonin and Martin. They're imagining reactions going on in little pools in a rock, little pores in a rock, and each of these little pores or crevices, molecules in one place can interact with each other, and then they move very slowly across the whole structure. So we now study a model like this, a lattice model where there can be many molecules per site, and things on one site can interact with one another, and there's some very slow hopping from one to the next. So this lattice model is representing something like that, and the nice thing now is that this is apples and apples, right? Because I can compare this lattice model with multiple strands per site with a protocell model with multiple strands per site, And the rules of replication inside one cell are the same as the rules for replication inside one site on the lattice. And so those two are directly comparable. What's different is the fact that in the cell model we have growth and division of cells. In the lattice model we have no growth and division, but we have diffusion between neighboring sites. So, while we do simulations of these things, we get different shapes of the error threshold curves, but qualitatively similar. You know, if there's no mutation, the red ones do well. You come to a point where mutation kills you, and this is the error threshold, and we want to know how do the error thresholds compare in these different models. Right. I got three curves here because there are three slightly different protocell models which I'm not going to explain. There are different ways of formulating these models. But all of these three are very similar, and they're protocell models. There's three curves here which are slightly different spatial models, and they're all much worse than the protocell models. And there's one in the middle which is our old version of one per site on the lattice. So this is the oranges, which we can't really compare, but these are two lots of apples, and what we say is protocells are much better than spatial models for two reasons. Because the error threshold, right, I should say, this is the error threshold. This is the maximum tolerated value of the error, and the error threshold is much higher for protocells than spatial models. And then this point here is the minimum replication rate necessary for survival. And the minimum replication rate for the protocells is less than it is for the spatial models, meaning that for both of those reasons, it's easier for replicators to survive in protocells. So, why do protocells work better? Because group selection works in protocells. Cells with good teams of molecules grow and divide, and they replace the slow-growing cells. And either we keep the cell constant, and when one cell multiplies, another one is removed, or we keep the number of strands limited, which then leads to an effective removal of the cells with no strands. So one way or another, there's group selection in protocells, which is not really happening in spatial models, because in spatial models, when we have good combination when we have a good team of molecules on one site, it fills up that site and stops itself replicating. It can only continue by diffusing molecules out of that site to its neighbors. So that mechanism works, but it works less well than the protocells. This is, how am I doing? This is just to say, so far I've been assuming that the rate of replication of the parasites was the same as the replication of the functional molecules. So the parasites were not adapted, they were just non-functional. Now I want to say, what if the parasites are adapted? They're adapted to be good templates, and they multiply faster than the functional molecules. So the blue curves are my old protocell and spatial case, where the replication rate of the parasite is the same as the polymerase, okay? Those two. And now I have a new version, the red and the pink, where the replication rate of the parasite is now twice as much as the replication rate of the polymerase. And so the answer is both the protocell and spatial models still survive even when you have adapted parasites, okay? So the parasites can adapt to be better than the polymerases themselves, and this mechanism of protocells or spatial models both save you from being killed by the parasites. But what happens then is that the error thresholds come down relative to the red curve is below the blue curve in both cases, right? But the difference between the red and the pink is actually larger than the difference between the blue and the two blues, okay? So when parasites are better, the advantage of protocells is larger than it was when they were neutral parasites. So I'm arguing that this advantage that we see for protocells is robust to different ways of formulating the model and to different rates of parasite multiplication. So what does all this mean then? It probably means that compartments like protocells came very early in evolution. So Damon Diemer envisaging replicating sequences inside vesicles that are formed when we have wetting and drying of of shallow pools in the presence of lipids, okay? So the environment provided by the lipids is good for polymerization. Maybe we get sandwich RNAs inside membranes, and it also creates vesicles, okay? So maybe they were present all along. Maybe the lipid membranes and the vesicles were present all along. Maybe the first replicators evolved inside cells. Okay, was that time? Okay, so the other thing I just want to say half a minute about is what we really need is something like this. We need multiple genes. So we don't just want one kind of replicator that replicates itself. We want that replicator then to be able to replicate other genes with different functions. So cells also have an advantage in that if you have molecules with different functions inside a cell, then group selection enables the survival of unlinked molecules with different functions. I'll finish there.
All right, so it looks like we have time for a few quick questions. So we'll start with Dave in the back.
In fact, you're wrong that the Smith surfaces, at least the reason you gave, things that move across surfaces don't need to dissociate to do so. They form bridging. There's a spot you can sit on, another spot you can sit on, and whatever attraction, often it can be a bridging attraction. They can sit on both of them for a moment and then move over. It's possible to find things, surfaces, such that they stay on the surface but move around the surface.
Right. So I see what you're saying. What you need in these molecules is very slow diffusion. Like you need the distance. Basically, they move by an amount of order their own size in their own lifetime, right? So in terms of a lattice, they can hop once to a neighboring lattice site in their own lifetime, and if they go faster than that, they end up being mixed, and then you lose the advantage of the clustering, right?
Activation is slower because they start feeling the binding to the new site before they completely leave the other one.
Yes.
So that the energetics for hopping are more favorable
Right.
than they are for dissociating.
Really what I'm trying to say is you need to have very slow diffusion, so you need to have strong binding, and if you have strong binding, you mess things up, right? You mess up the template.
Oxygen platinum jitter right across the surface.
Okay. Maybe we can quickly do one more question in the front.
Thanks. I just wanted to ask about, so in terms of the cooperation barrier, in sort of evolved life, we see various policing mechanisms and sort of suppression of cheaters. I just wondered if you had any comment on how that might come into play.
Okay, I know that policing comes up when you're talking about evolutionary cooperation in organisms. What does a policing molecule, tell me what a police molecule is. I don't know what it is.
Well, so apart from just doing group selection of protocells, the other way of suppressing molecular cheaters would be to have some sort of chemical method of suppressing the parasites within.
Something that comes to mind there is this idea that you can have tags on molecules which indicate that this is a functional molecule and it must be present in order for the polymerase to replicate this sequence, right? So that can eliminate parasites which don't have the tag, but doesn't eliminate mutations occurring in the functional molecule such that the functional bit is no longer functional and the tag is still present. So there's a little bit along the lines that you're talking about, but I don't think it saves you from there is a problem here of parasites which doesn't go away by that. Yeah.
All right, well, thank you, Paul. So, yes, thank you.